So first of all, thanks to the invitation. I'm a theoretical physicist. I graduated in theoretical physics, and once you graduate in theoretical physics, you're a theoretical physicist forever. And but today I will talk about computational model for neuropathologies. So one of the most recurring quotes in our lab is the famous incipit by Anna Karenina, happy families are all alike. Every unhappy family is unhappy in its own way. Why is this? Why do we talk about it? Because when we compare the results for healthy patients and the results for patients with some kind of disorders, we always find more variability in the second case. So there's everything, every perspective you look at it, you find more variability in patients with disorders or in data acquired from dysfunctional nervous systems than from the healthy nervous system. This is not because you have many possible disorders. Even within a single disorder, as was beautifully described in a book that I strongly recommend, Awakenings by Oliver Sacks, in which he was one among the first to test dopamine treatment to people having some kind of Parkinsonism, lethargic Parkinsonism, but the therapy is still there. And he was observing really, I would say, wild variety of responses to exactly the same dose of drug to people who had exactly the same disorder. So we must cease to regard all patients as replicas, okay? And honor each one with individual reaction and propensities. And this will improve our understanding of the phenomenon and also our clinical approach. This is really important. I think that there's a reason why this wild variety was observed in Parkinson, because Parkinson involves a particular region of the nervous system called the basal ganglia. So the basal ganglia are at the base of so this is the cortex, this is what you think is the emoticon of the brain, right? While at the bottom you can find all the different structures of the basal ganglia. You can see that they are quite diverse and sparse, but being people who study physics, we do not care much about the anatomy, we do care about the functional network. And the problem is that basal ganglia are a network of networks within a network. And I literally mean it. So the point is that when the cortex sends instruction about the movement, let's dance, to the spinal cord, then what happens is that it also sends an efferent copy to the basal ganglia, which are this area here within the blue box, okay, which sends back some feedback like stop or increase, accelerate, slow down, okay, through the thalamus. So the big network is cortex, basal ganglia, thalamus. Then the basal ganglia themselves, they are composed by different nuclei, striatum, globus pallidus, pars externa, interna, substantia nigra, subthalamic nucleus, and so on. Some of them, so this is a network within the network, but some of them have an internal structure, in particular the striatum. So you can see how in such a, we have a complex network, so it's not surprising that changing one single neural transmission, in particular, the transmission of dopamine from the substantia nigra to the striatum can cause dramatic effects. For instance, Parkinson's disease. So what you can see here, basically, to make the long story short, is that the same molecule, so the same neurotransmitter, dopamine, has on one side an excitatory effect and on one side an inhibitory effect. So everything is balanced until the dopamine is there, but then as soon as the dopamine decreases or disappears, then what you have is an alteration between a crucial element of our brain, which is the balance between excitation and inhibition, and this reverberates to everything. At the first order, you can say that the patient slows down. So you have a prevalence of the inhibitory system, but it's a complex system. So it is not the whole story, because then you also have pathological oscillations. When we think about Parkinson, we do think about tremor, not only about people slowing down. How can a pathological oscillation arise from an hyper-inhibited network? It's one of the challenges, okay? And then there are even effects, I won't discuss them there because there's no time, but also effects on the cognitive aspects, okay? So it's not just a motor issue. And the first thing before modeling is, as was said correctly in the last talk, is to collect data, and it's not always easy. You don't really find many occasions to collect data from awake human beings. But the strange thing about Parkinson is that there's this very effective therapy called deep brain stimulation that is in the clinical practice since the 90s. So they're stimulating the part of the brain, the basal ganglia, which are deep, so they have to go through the whole cortex, basically. And in this way, they can ameliorate some motor symptoms of Parkinson. Is quite effective. And at the same time, it opens for us the possibility to record from there. So we're recording the activity of deep structures in the human brain of awake subjects. And this is incredible. It gives us the possibility to understand a lot of things. So, for instance, in one research I did in collaboration with Würzburg University, we were trying to understand the origin of tremor, okay? The origin of tremor and how do these oscillations that are way faster than tremor oscillations can play a role in tremor. So what you see here is the power spectral density of the network activity recorded in these basal ganglia, and we found that was neurologists do love these big bands, alpha, beta, delta. They're not even in order for some reason, and of frequency. But actually, sometimes they're sometimes useful. Sometimes, as all conventions, they're not useful anymore. So we found that inside what was defined as a global beta oscillation, there were actually two different sub-oscillations carrying different information, and one of them was about the onset of the movement, and the other one was about how to perform the movement. So we refined the relationship between movement and neural activity. Then there's also this great problem that is called the freezing of gait. So Parkinsonian patients can walk, but then slowly, but can walk. If they find an obstacle, the slow down, because you have to take care, for instance, even going down a single step or avoiding hitting a person in a crowded situation, the slow down message is so amplified because of the lack of dopamine that they actually freeze, and they cannot restart moving anymore until you physically push them, okay? So what we were studying this, and again, we used the tool of spectral information to find that in this case, it was not much about an increase in the power of this pathological oscillation, but a slight change in the peak frequency. Again, this is from human patients in the University of Würzburg. Then we said, okay, we collected some data, we want to start, the picture is not clear enough, we want to build the first model. And so, as usually do, the best thing to do is to learn from people that know more than you. So we went to the Karolinska Institute in Stockholm, where they have strong studies, and we developed our first model network of the basal ganglia. So each one of them is a single neuron, spiking neuron. You see there's a very complex interaction. We won't go into detail, but the interesting thing was that we were able to find to reproduce the pathological oscillations, and we found also that they were correlated to the kind of input that the striatum received, which was cool, but not giving enough insight. So why dopamine, where this oscillation comes from exactly. So the second step, now we learned, so we don't need the Karolinska Institute anymore. We did it between us and the Department of Physics here in the context of the beautiful complex systems course that it's held here. So this was a shared student. And the first thing, so you can find all the details in the poster there, so ask the student, he knows more than me. But then the basic idea was that in this complex, very complex network, we were able, Andrea Ortone together with Alberto Vergani, to find two oscillators, so two clean oscillators, and both had a specific resonance exactly within the range of the pathological oscillations. But the point was then to study their interplay. So we went on and we found that depending on the level of interplay, then they were going toward an average frequency, and even more important, the oscillation were more and more robust. And finally, instead of generally altering the input to the network. We really manipulated, because in this case we had explicit equations about that, the depletion of dopamine that is observed in Parkinsonian patients. And once again, we found that not only it is supported the synchronization, but made even more stronger and robust to external stimuli. So it's oscillating no matter what, that if you want, in a system that requires to be flexible like the human brain, this is, if you want, the definition of pathological oscillation. So this is what we have done with the Parkinson system. But then, before Parkinson, there was the visual cortex. So the first model that I studied was the visual cortex, and it's also the classical benchmark for all the studies in computational neuroscience, starting from the pioneering studies of Hubel and Wiesel. And the study that I did in my youth basically was, again, a model of a patch of visual cortex. And what we found was that there was an internal resonance that was modulated by the external stimuli. So an increasing level of contrast in the vision didn't correspond simply to an increase of activity in the visual cortex, but rather to the emergence of a collective oscillations. And I really mean collective in the sense that no single neuron is oscillating, but the whole network is. It's called weak synchronization. But then we want, again, the point is that we don't think anymore that it's time to just study the nervous system. It's time to change it, or at least to fix it, to replace missing functions and to fix those that are not working. In this case, in the visual cortex, you don't get many occasions to collect data from the human brain of people having disturbances in the visual cortex. So we had to use mouse models in collaboration with the CNR here in Pisa. And the first thing was that, okay, I thought, okay, I have a model that is working from primates and humans. I mean, we are more evolute than mice, so it would be super easy to apply them to mice. Actually, it was not, because the little bastards have two visual pathways, one for when they are in the dark underground, and one when they are outside. So outside, so for high contrast conditions, my model was working. But then they also have a second pathway for when they are in low contrast and they become sensitive to light. So it took a whole year of the project just to adapt the primate model to the mouse model. But then once we did it, this was published in the end, thanks to Niccolo Meneghetti. And then we moved to pathology again. Now, this is interesting because the visual cortex is not as complex as the basal ganglia. Actually, you can even, we're doing spiking neural models to understand better what's going on, but to some extent, you can even do a mean field approximation of the dynamics there. So, but again, pathologies are very complex. So in this case, complexity is not in the structure. So in Parkinson, we had a complex structure and a simple modification, dopamine depletion. In this case, we have a more simple network, visual cortex, relatively coherent, then you have this different receptive fields, but they're quite similar, and very complex pathologies. So a single pathology, actually, I hope this is the only time that I'm studying a pathology that I suffer from. So when we studied migraine, we found that the possible chemical modifications caused by migraine were virtually endless. So again, the problem of all of us when we are doing models is, which elements do I have to include in my model? So we started with a always follow Occam-Razor principle. So we started the first one. Maybe we just can explain what happens in migraine by looking at the modulation in the cortex. No, we have to take into account the modification in the cortex and in the thalamus. Quite almost there, but then we have to take into account that these modifications are different for excitatory and inhibitory currents. So just once we did this, we were able to explain some interesting facts. For example, the fact that the resonance is still there, but when the resonance in modulation to the, depending on the visual stimuli, but moves to quite higher frequencies. So the resonation moves from, the resonance moved from 30 Hz to almost 90, and we think that this might play a role to the hypersensitivity to light that people have in migraine, okay? Then we want to explore, and this again was part of the study of Niccolo Meneghetti. Then again, since this collaboration with the physics department is providing us with a lot of young blood willing to explore the world of computation neuroscience, we were wondering, so in this case, we do have, so migraine depends on an increase at the cortical level of excitation. But then we know that epilepsy comes from a lack of inhibition. So minus per minus equal plus. How comes that they're so widely different? Okay, so an increase in excitation leads to migraine, which is bad, and while a decrease in inhibition leads to epilepsy, that is a possible life-threatening condition. And so together with Federico Dalba, who's there, we are studying so the difference in the dynamics of, in particular, of the balance between excitation and inhibition when we increase AMPA versus when we decrease GABA. And basically, I think it's a complex study, but of course you can see that you do have some transition here where for very low GABA that you don't observe for very high AMPA. We will keep tuned a bit, and we will update you on why it is actually happening. But also in our models, we find that the minus per minus is not plus in complex neural systems. So then, again, with another student of physics, Dario Milea, we are trying to take into account the fact that to add another piece of realism, which is actually there are two kinds of interneurons, and it seems that migraine is affecting one of them more than the others. But as you can see here, he's producing very nice results, but I wanted to reproduce this methodological figure from his thesis. So as soon as you add an extra element in the network, the complexity of the network increases non-linearly, of course. You have to take into account the self-recurrent inhibition, you have to take into account the interplay with all the other things. So it's quite a long and complex work. Finally, together with Angelo La Sala, who's also there, we went instead more microscopic. So we actually won, and it's a big responsibility, a grant to study yet another kind of pathology on the visual cortex, that is glioma. So the glia is a substrate of cells that are not conducing energy, but actually are supporting with energy, not conducing electricity, but are supporting with some energy and modulation the nervous system. This forms a tripartite model, taking into account the fact that neurons interact with neurons, but neurons interact with glia, and glia interacts with neurons. And in the end, it's really interesting because we find out that as soon as we introduce an interaction between an open loop interaction, you have some changes in the spectrum, but in the spectrum of the neural activity, but they become dramatic once you take into account the closed loop. So the interplay with the glia is actually shaping the resonances of the brain. Again, this is work is going on, and but we find that so they play a role in determining the resonances of the brain. So probably when the glioma kills the glia, these resonances go wrong. That's our idea right now. And in this case is one of the cases in which we can use the mean field approximation if you're as smart as Angelo, and we found very good agreements with the simulation, thinking that we maybe grasp the basic idea of the problem. So, last but not least, what I've shown you so far are the large-scale simulations. So when you want to take into account specific circuits like the basal ganglia or one patch of the visual cortex, then you can do actually the model taking into account virtually every single neuron and the interplay of every single neuron with every other single neuron. We call them spiky neural networks. But in some cases, when you have to deal with pathologies that involve the whole brain or reverberate over the whole brain, then it's not even just useless, but it might also be misleading looking at the single neuron firing. So we used some coarse models, okay, in which every area is computed as a neural mass model, so a set of equations for each area rather than a set of equations for each single neuron. And again, it is interesting because we found already there in the public repository of European community a good model for the healthy brain. That was good because we could not start by developing by herself the model of the healthy brain. But there was no model for Alzheimer. In this case, this is not a project funded by Regione Toscana about the study of Alzheimer. So this is it's really representative of the current state of computation neuroscience. We don't know everything about how the healthy brain behaves, but we know we have reached the critical stage. What we do not know is how pathologies unfold. And it's a pity because it's really interesting and it's what really affects in the end the life of the people. So coming back to the thesis of Lorenzo, who's there, and there's also a poster about it there. So for the mathematical detail, ask Lorenzo and Alberto. So the idea is that we implemented similar to what we have done for migraine. Our idea is the same. So we know from neurophysiology studies, from histological studies, whatever you want, what is the effect of these pathologies at the small level, at the micro or scopic level. So in this case, a depletion of inhibitory neurons and an hyperexcitability and the loss of some connections. The interesting thing was to understand how these things reverberate. So this is an experiment that, I mean, people cannot, neurologists cannot switch on and off the different aspects of Alzheimer to see how this reverberates on EEG. We can. So what Lorenzo did in his thesis, he just graduated last week, congratulations, was both to reproduce the changes at the level of the single patch of cortex. In particular, again, it's all about resonances. You see, it's not more about the brain is active or non-active. The point is really the spectrum of the brain what matters, according to me. So in this case, we found we were able to reproduce a change in spectrum that was observed in Alzheimer patient. And even more interesting, that we found that there was a particular index of graph theory, Randich index, that was describing the evolution of Alzheimer. But what was really interesting is that in this case, we find that the evolution is probably non-linear. I mean, we are a complex system, Congress, linear functions are boring. So while in this case, it seems that due to an interplay of different synchronous factors, in the first stage of Alzheimer, we do have, there is a change, there is a decrease in the Randich index. Okay, so even if it sounds strange, you have a kind of reinforcement due to compensatory effect of the connectivity, and then the connectivity is completely lost only for prodromic Alzheimer, for full Alzheimer. So if you want to look for early stage of Alzheimer, that's the counterintuitive but very interesting conclusion, you don't have to look for a mild depletion of connectivity. No, you have to look for an increase in connectivity. So we think this is a very interesting result. That again comes from the interplay from neurology, neuroscience, and physics of complex systems. That's basically it. I want to keep it short. So why study computation model of neural pathologies? The first thing is that complex paths await you once you are outside the stable states. So it's really interesting. You appreciate the complexity of the model once you are outside the stable state, where if you are nothing happens, everything is in balance. And then you find out if your model is sufficient. And then the point is that this put your model under stress, because then you want to, the point that you really understood what's going on if your model is also able to explain what happens when you have parameters that are changed or biological variables that are changed in reality outside the basin of the attraction of the stable state. Then, even if it sounds really strange, sometimes you only have access to pathological data. So we don't have access to recording from the subthalamic nucleus or the basal ganglia from healthy patients, because we respect human beings and we don't do holes in the skull unless it's really necessary. And so the point is right now, try to understand what of we are recording from the basal ganglia is actually due to Parkinson, and what is actually we can transfer from other, like for instance, doing models of Parkinson that cover also EEG, as Maria Laura is doing right now. And in this case, maybe we can have a non-invasive proxy of the invasive data that we are collecting. And the most important thing, is that the time is now. So we know we have studied the nervous system forever, neurophysiology more than 100 years, computation neuroscience roughly from since I was born. The point is that we do know enough, not everything, but enough of the brain, of the healthy brain, that it's time to go and face the real problems and try to understand pathologies and try to make neuroengineering and medicine and neurology closer to hard science for a mutual benefit. Thank you.
May I ask a question?
Thank you very much, Roberto. Questions?
Can I ask a question?
Yeah, Bruce?
Yes. What are your models sufficiently sophisticated to discuss something like the 40 hertz flickering lights and flickering sounds that have been used in mouse models to reduce the plaque buildup in early Alzheimer's disease by up to 60%?
I didn't. So to use for Alzheimer's disease and for what other? What was the other thing that was reduced?
Oh, the plaque buildup in early onset.
Plaque buildup. Okay. So actually, okay, this was the last slide, and thanks to the whole lab. And so, but first I'll answer the question. So right now for Alzheimer's disease, we are working. It's another case. Sorry, I didn't explicitly say this, but we are back to human data. So it is a project with the Tuscany region in which we are analyzing non-invasively, so recording EEG in patients with full Alzheimer and and prodromic states of Alzheimer, like mild cognitive impairment and subjective cognitive decline. Now, this is a good question. So we didn't try, but I think in this case, given the fact that we can exploit, in this case, the fact that we have a model that we can apply to humans, of which we are monitoring the clinical state. So I think a good next step for our work, once that, actually, yeah, perfectly, it's the first thing that we do once we have a pathological model, is to develop in silico test of possible therapies. So I think it will, thank you for the suggestion. It will be very interesting to see, to provide actually, so to human beings, even more interesting, to provide this particular kind of stimulation and to see if these are correspond also in our patients to a clinical improvement, and try to see if the model we just developed is able, I mean, the important point for us is, I mean, to understand the underlying mechanism. So we didn't try, but thanks for the survey, for the suggestion.
Other questions? I know, well, people know that one of the first symptoms of Parkinson has to do with sleep disorders. And so I was asking if the decrease of GABA and things like that work with your model and may impede a good sleep.
So yeah, this is really interesting. So, but again, in this case, we are right now collecting data, because it's not easy to collect data from sleeping patients, because the deep brain stimulation device, so for decades has been used to record only right after the implant or at battery recharge. So it's just since March 2020 that selected hospitals, but among which both the Careggi Hospital and the Würzburg Hospital we collaborate with, have access to one device that is called Percept from Medtronic, that enables us also to record 24/7. So we have now, but then March 2020 wasn't the right time to start the clinical studies. There were a lot of problems. So we start having internal data from what is actually going on at the level of sleep right now. And this is interesting because, so we do see some differences, but we are still on the stage of collecting data. So I think probably, so fluctuations in dopamine are, there's an amazing book that is just out now, Hidden Life of Basal Ganglia by a guy Bergman, and shows how the fluctuations of dopamine are all day long, basically, then all lifelong. So the reason why we are more prone to explore and usually more conservative when we are young and more conservative when we are old is simply that the dopamine is declining. There's nothing philosophical in it from my materialistic point of view. And then again, dopamine is one of the several chemicals that are fluctuating between awake and sleep cycle. And actually, the adaptive deep brain stimulation, so far, the only of widespread use is to change the level of stimulation, making it slower, less intense during sleep. So all the data, right now in this case, we are at the data analysis stage, but we think this is very interesting as well.